Hello everyone, welcome to Science Express. Today our topic is nucleic acid. But in this lecture, we will be discuss about the structure of DNA. Now start the lecture. The DNA, it is an acidic substance and it present inside the nucleus. DNA was first identified by the scientist Frederick Mixture in the year of 1869, but he named it as a nuclein. Okay. The term DNA was given by Jacqueries and it is a double helix model that was proposed by James Watson and Francis Crick in the year of 1953 based on the X-ray diffraction data proposed by Wilkins and Rosalind Franklin. Okay, so term DNA was given by the Jacqueries and double helix model was proposed by the scientists Watson and Crick on the basis of X-ray diffraction data proposed by Wilkins and Rosalind Franklin. That is the history of DNA. Okay, now start the basic structure of DNA. DNA is a long chain of polynucleotide. The length of DNA is usually defined as a number of nucleotides present on it. Okay, now E. coli, that one is a bacteria, has near about 4.6 into 10 to the power 6 base pair and haploid content of human DNA is about 3.3 into 10 to the power 9 base pair present on it. Now here we can see that structure of polynucleotide chain. Nucleotide, it has three component: nitrogenous base, pentose sugar, that type is ribose, and deoxyribose. Ribose mainly present on the RNA and deoxyribose present in the DNA, and phosphate group. Okay, nitrogen base are four types actually: A, T, G, C, and in case of RNA, T will be replaced by the U. A for adenine, T for thymine, G for guanine, C for cytosine, and in case of RNA, T will be replaced by the U. U means uracil. Okay, so three component mainly present. In the picture we can see that one is the nitrogen base, then pentose sugar, okay, and then phosphate group. Three combination of this structure is called nucleotide. Now there are two type of nitrogen base. One type is the purine, another type is the pyrimidine. Purine, this is mainly A and G, that means adenine and guanine, and pyrimidine C, U and T. Okay, C for cytosine, U for uracil, and T for thymine. So that is the picture of adenine and guanine. These are nitrogenous base, and adenine and guanine, it is a double cyclic ring like structure and it is a combination of carbon and nitrogen ring. Now cytosine is common both DNA and RNA, but thymine is present in the DNA and uracil present in RNA at the place of thymine. So these are the pyrimidine nitrogenous base: thymine, cytosine and uracil. Okay, so these are single ring structure. Now discuss about the pentose sugar. Pentose sugar mainly two type. One is the deoxyribose sugar, another one is the ribose sugar. Pentose sugar means in a ring mainly 5 carbon present, but the ring formation by the 4 carbon. Okay, total number of carbon is the 5 carbon, but the ring carbon is 1, 2, 3 and 4, and 5 carbon is the hanging, this position oxygen occupied. So the ring name is the furanose. Okay, furanose, and total number of carbon is here 5, so that one is a pentose sugar. Clear? So in case of the DNA, name of the pentose sugar is the deoxyribose sugar, and in case of the RNA, name of the ribose sugar is the RNA ribose sugar mainly. Okay, so in the picture we can see that the deoxyribose sugar and ribose sugar difference on the second number carbon. Okay, this is the third and fourth and fifth carbon, first, second, third, fourth and fifth carbon. In the second number carbon we can see that In case of the ribose sugar, there is a one hydroxyl group present, but in case of the deoxyribose sugar, there is a no hydroxyl group present. Okay, so name it is a deoxyribose sugar. Now, a nitrogenous base is linked to hydroxyl of first carbon pentose sugar through N-glycosidic linkage to nucleoside. That means nucleoside means when pentose sugar plus nitrogen base. These two things are together called nucleoside. When phosphate group attach here, that means phosphate group attach on the pentose sugar and nitrogen base, that three combination is called nucleotide. When phosphate group is attached, is linked to hydroxyl of the 5 carbon of the nucleoside through phosphoester linkage, and corresponding nucleotide is formed. Clear? Now, two nucleotides are linked together 3,5 phosphoester linkage to form a dinucleotide. When two nucleotides are attached in a chain, that name is the dinucleotide. More nucleotide can be joined to form polynucleotide. Okay, so DNA is a long chain of polynucleotide. Clear? Okay. Now, a polymer thus formed has at one end a free phosphate moiety, that is the 5' end of the sugar, which is the referred to as the 5' end of the polynucleotide chain. Similarly, at the other end of the polymer, the sugar has free hydroxyl group, which is referred to as the 3' end of the polynucleotide chain. The backbone of polynucleotide chain is formed due to sugar and phosphate, and the nitrogenous base is linked to sugar moiety project from the backbone. Okay, in the picture we can easily understand. Okay, these are the structure of nucleotide. Okay, in the nucleotide, one nitrogen base, here guanine and pentose sugar and phosphate, these one are nucleotide. Okay, DNA is a long chain of nucleotide. Okay, 1 2 3 4 5 6, here is a 6 nucleotide present. Clear? Okay, nucleotide, in a nucleotide, pentose sugar attached to the nitrogen base, the bond is called glycosidic linkage. Okay, and pentose sugar attached to the phosphate, here is the phosphoester bond. Clear? Okay. Now, in this chain we can see that both chains are arranged in a anti-parallel fashion. Clear? That means in the first chain we can see that here the 5' end, that means phosphate end, is free, and the ending position hydroxyl group is the free. Clear? So here we marking here 5' end to 3' end. Clear? But in another chain, here we can see that that position is the hydroxyl one. Okay, and that position is the phosphate group. Clear? So here we can see that the hydroxyl group, its position is the 3, and here phosphate position is the 5. So the orientation of the two nucleotide chain is the 5 to 3' and 3 to 5'. Clear? Here we discuss about the Arvin-Chargaff rule. What is Chargaff rule? In a double strand DNA, the ratio between adenine and thymine and guanine and cytosine are constant and equals 1. That means total amount of purine equals to total amount of pyrimidine. Clear? So we can write here A plus G by T plus C equals to 1. Clear? Okay, that is the Chargaff rule. Okay, so it is applicable only dsDNA, that means double standard DNA. Clear? Now discuss about the base pairing rule. The base pairing rule confer a very unique property of the polynucleotide chain. They are said to be complementary to each other, and therefore if the sequence of the base in one strand is known, then the sequence of the other strand can be predicted. Okay, in a chain, the number of A, that means adenine, present, how much amount, then the amount of T will be same. Clear? A, number of A equals to number of T. Clear? Suppose assume that A is the 20%, so T will be automatically 20%. Clear? And rest of the 60% will be divided into among the G and C. So G will be 30% and C will be 30%. Clear? So total amount of purine, let's suppose example 30 plus 20 equals to total amount of pyrimidine, that means 30 plus 20. Clear? So base pairing rule is total amount of adenine equals to total amount of thymine and total amount of guanine equals to total amount of cytosine. Clear? Now discuss about the salient features of double helix structure of DNA. It is made of two polynucleotide chain where the backbone is constituted by the sugar phosphate and the base projected inside. Clear? Now the two chain have anti-parallel polarity, already discussed. It means the chain has the polarity 5 to 3' end and other has 3 to 5' end. Okay, so both chain are arranged in a anti-parallel fashion. 5' means the one chain is the here 5' phosphate is free and another end is the hydroxyl group, that means 3 end is free, and opposite chain, another chain is the 3', that means hydroxyl end is free, and here 5' is the free, that means phosphate is free. So they are arranged in a anti-parallel manner. Now the base of two strands are paired with hydrogen bond forming the base pairs. Adenine is bonded with the thymine of 2 hydrogen bond and guanine is bonded with cytosine with 3 hydrogen bond and vice versa. That means adenine pair with the thymine by the 2 hydrogen bond and guanine pair with the cytosine 3 hydrogen bond. Okay. Now and also vice versa means thymine also paired with the adenine with the 2 hydrogen bond and cytosine also paired with the guanine with the 3 hydrogen bond. Okay. Now discuss about the DNA double helix structure model. That one is also called BDNA. Okay. So Watson-Crick DNA is called BDNA. We discuss here BDNA. There is another mainly 5 type of DNA. One is the ADNA, BDNA, cDNA, ddNA and zDNA. Okay. So here we discuss only BDNA. The two chains are coiled in a right-handed fashion. Clear? So helix is a right-handed fashion. Clear? In case of the BDNA, ADNA, cDNA and ddNA. But in case of the zDNA, the helix is the left-handed fashion. Okay. The pitch of the helix is 3.4 nanometer and are roughly 10 base pair in each turn. Clear? So length of the DNA in case of the pitch of the helix is near about the 3.4 nanometer and are roughly 10 base pair present each turn. Each turn means, so in the picture we can see that there is, in case of DNA, there is, here we can see that two groups. That one is called major group and that portion is called minor group. Okay, one major group to another major group, that length is called one turn. Okay, in this turn the length of DNA is the 3.4 nanometer or we can call it 34 Armstrong. Okay, and here in case of the BDNA the number of base is the 10 base pair present in the 34 Armstrong. So length of the each base pair or distance between the two base pairs is 3.4 Armstrong. Okay, or is about 0.34 nanometer. Clear? Now the plane of one base pair stack over the other in a double helix. This is in addition to hydrogen bond confer stability to the helical structure. Okay. Now discuss about the packaging of DNA. Packaging means how DNA is present inside the nucleus. In a prokaryotics, they do not have identified nucleus and DNA is not scattered throughout the cell. Okay, that means scattered throughout the cytoplasm. DNA is held with some protein in a region termed as nucleoid. Okay, nucleoid means where DNA is present without nucleus. Okay, the DNA in nucleoid is organized in a large loop held by protein. In eukaryotes, this organization is much more complex. There is a set of positively charged basic protein called histone. So, in case of the eukaryotes, there is a protein present. That name is the protein is histone, and mainly the protein is positively charged. Okay, and the histone nature is, protein nature is the basic in nature. So, it is made up by the basic amino acid, name of one lysine and arginine. Histone are organized to form a unit of 8 molecule called histone octamer. So in the picture we can see that here is a 4 type of histone present: H2A, H2B, H3 and H4. Okay, they are present in into 2 number 2 and form octamer. So that one is the octamer. Clear? Histone octamer. Okay, so here we can write that H2A, H2B, H3 and H4. The 4 histone protein, they are present in a double number into 2. So number is the 8 and they from histone octamer. Okay, and DNA wrapped around the histone protein. Clear? DNA is mainly negatively charged and acidic in nature, and histone protein is basic in nature and it is positively charged. Okay, histone protein, another one is histone protein present, that name is the H1. Okay, and in case of bird, the H1 is replaced by the H5. Okay, let's discuss about the H1 function. Here we can see that one is the histone H1 protein. Okay, histone H1 not involved in the octamer, they are present outside this core region. Okay, now here we can see that nucleosome, the negativity charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. Okay, so this total structure is called core DNA, linker DNA, and histone octamer is called nucleosome. The typical nucleosome contain near about 200 base pair of DNA helix. Okay, so here involve 200 base pairs. Clear? Histone 1 here help actually binding or mainly tightening of the octamer or DNA. Now discuss about the chromatin. Nucleosome constitute the repeating unit of a structure of nucleus is called chromatin. That one is a thread-like stained body seen in the nucleus. The nucleosome in chromatin are seen as beads on string structure when viewed under the electron microscope. Okay, so chromatin, it is a thread-like stained body seen in the inside the nucleus, and nucleosome in chromatin are seen in a beads on string like structure. Okay, under the electron microscope. So in this picture we can see that one is the beads on a string nucleosome array. And how we can see that here, how DNA is packaged. Okay, and this form is transferred into the forming a structure, that is chromosome. So the beads on string structure in chromatin is packaged to form chromatin fiber that are further coiled and condensed to form chromosome. Okay, now discuss two type of mainly chromatin are present. One is the euchromatin, another one is the heterochromatin. In typical nucleus, some region of chromatin are loosely packed. They are referred to as a euchromatin, and the chromatin that is more densely packed, they are stained dark, are called heterochromatin. So euchromatin loosely packed and heterochromatin is the densely packed. Okay, on the basis of the packaging we divided them two category. Eukaryotic, in case of the euchromatin, they are transcriptionally active. That means they can synthesize mRNA and protein also. Okay, actually they produce the gene, functional gene resultant. Okay, in case of heterochromatin, they are inactive, so there is a no product is produced, so gene is inactive in nature. Example, heterochromatin is the X chromosome that are present in a female body as a bar body. Clear? So bar body is a example of heterochromatin. In the picture we can see that in a deeper density color, that is the heterochromatin, that one is the silent, so there is a no transcription occur, and in case of the eukaryotic they are loosely packed. Okay, so there is a transcription occur, their active site for the protein synthesis. Okay, thank you. Please do like, comment, share and subscribe Science Express.